Hello everyone, and welcome to our latest Phlebotomy Study Log. This video focuses on the chapter on special collections and procedures. And I have 25 questions prepared for us. For each one, hit that pause button and see if you can figure it out before I go over the answer. I hope this helps. Let's get started. Number 1. List 10 factors that influence blood composition. Blood composition is influenced by age, altitude, dehydration, environment, gender, pregnancy, stress, diet, diurnal variation, drugs, exercise, body position, and smoking. Number 2. What 3 things are timed specimens most often used to monitor? Timed specimens are most often used to monitor medication levels, changes in a patient's condition, and normal diurnal variation in blood levels at different times of the day. Number 3. What is an OGTT? GDT is the acronym for glucose tolerance test. It is a test used to screen for diabetes mellitus and other disorders of carbohydrate metabolism. Number 4. What is TDM, and why would this be ordered? TDM means therapeutic drug monitoring. It is often used to adjust drug dosing in patients. Number 5. Why are BCs ordered? Blood cultures are ordered to test for the presence of microorganisms in the blood. Number 6. What collection technique is critical for meaningful BC results? Aseptic collection technique and drawing the correct volume are critical for meaningful BC results. Number 7. Describe the screening process a potential blood donor goes through. A potential blood donor must 1. Register by providing identifying information and written consent. 2. Interview with a trained interviewer and provide a medical history. And 3. Submit to a physical examination, which includes hemoglobin testing. Number 8. What special handling procedure is necessary for cold agglutinins samples? Cold agglutinins must be kept warm until the serum is separated from the cells. They can be wrapped in an activated heel warmer packet or placed in the incubator. Number 9. List 5 tests that require transport on ice. Tests that require the sample be transported on ice include ABGs, ammonia, lactic acid, pyruvate, glucagon, gastrin, adrenocorticotropic hormone, and parathyroid hormone. Number 10. Explain COC. Chain of custody, COC, is a protocol that ensures that a sample is always in the custody of a person legally entrusted to be in control of it. The chain begins with patient identification and continues through every step of the collection and testing process. COC documentation includes special containers, seals, and forms, as well as the date, time, and identification of the handler. Number 11. Describe the important features of blood alcohol testing. For blood alcohol testing, the site must not be cleaned with alcohol, as this will falsely elevate the result. Use soap, water, and gauze or another non-alcoholic antiseptic solution instead. Tubes must be filled as full as the vacuum allows to minimize the escape of alcohol from the specimen into the space above, and the specimen should not be uncapped, because that also allows alcohol to escape and compromises the integrity of the sample before testing. Number 12. Explain the procedure for preparing a blood smear. To prepare a blood smear, place a drop of blood on a clean slide 1/2 to 1 inch from the end, centered between the 2 slides. Place the spreader onto the first slide at a 25 to 30 degree angle and draw it back to just contact the blood drop, and then move the spreader forward in one continuous movement to the end of the slide. The blood will be drawn along over the slide. Finally, dry and label the slide, using a pencil on the frosted end. Number 13. Describe the procedure for preparing a thick smear to test for malaria. To prepare a thick malaria smear, place a large drop of blood on a slide, Spread it out to about the size of a dime, and let the sample dry for at least 2 hours. Number 14. List the disorders for which therapeutic phlebotomy is used. Define the diseases based on the word roots, prefixes, suffixes, or a combination of these. Therapeutic phlebotomy is often used in the treatment of polycythemia. The prefix poly means excessive or a lot, the root word cyte means cell, and the suffix emia means blood. Thus, polycythemia is excessive red blood cell production. Therapeutic phlebotomy is also used for hemochromatosis. Heme refers to the blood and chroma, to color. In hemochromatosis, the skin takes on a bronze color because of excess iron in the blood. Number 15. Time specimens are most frequently collected to monitor. Number 15. Medication levels. Number 16. Hyperglycemia means increased glucose. Number 17. For a 3-hour OGTT, how many blood samples will be collected? 4. Number 18. Within what time period should a patient drink the glucose solution required for a OGTT? 5 minutes. Number 19. BC, or blood culture collection, involves an aerobic and an anaerobic sample. Number 19. Therapeutic phlebotomies are commonly performed on patients with hemochromatosis and polycythemia. Number 21. To collect a cold agglutinin sample. Warm the sample tube for 30 minutes before collection. Number 22. Which specimen requires chilling during transfer to the laboratory? Ammonia. Number 23. When making smears for malaria. Prepare 2 to 3 regular smears and 1 thick smear. Number 24. It is necessary for a patient to fast for 12 hours before 3-hour OG. Number 25. Which of the following specimens is not sensitive to light? Lactic acid.
Thank you for watching. Don't forget to subscribe to our channel for more content.